Hello, everyone. So thank you for tuning in. My name is Associate Professor Dr. Muhammad Razif Shahril. I'm teaching nutrition in University Kebangsaan Malaysia. So today we'll talk about nutrition and epidemiology and also its relationship with the policy development. So before we move on, let's look at the definition of epidemiology. So what is epidemiology? Epidemiology is a study of the distribution and also the determinants of health-related states or events, including diseases like cancer, heart disease, hypertension, diabetes mellitus, so on, in the human population, and the application of this study to the prevention and control of diseases and also health problems. So it's actually looking at who gets the disease and why they are getting the disease. So an epidemiologist study sick and also well people, okay? So to determine the crucial difference between those who get the disease and those who are spared from the disease. So some people describe it as a study of epidemics. So what is epidemic? So epidemic occurs when there is a significantly more cases of the same disease than the past experience could have predicted. So there are various methods that can be used to carry out epidemiological investigations. First one is surveillance and descriptive studies that can be used to study the distribution of the disease, and also analytical studies that are used to study the determinants of the disease. So the study of distribution and determinants or exposure of disease or health. So this is initially used to investigate, control, and prevent epidemics of infectious diseases. But now it's also applied to studies of injuries, chronic disease, and also social problems. So exposure and also the health outcomes. So in nutritional epidemiology, it studies the frequency, the distribution, and also the nutritional determinants of disease, and also other health-related conditions in human populations. So the exposure we are talking about is the characteristics of an agent that a person comes in contact with that may be related to disease risk. So in nutrition, the nutritional exposure includes diets, including nutrients, food items, food groups, and also dietary patterns. We also look at body size as an exposure and also physical activity. So what is meant by frequency? So frequency is the number of times an event occurs. The normal question will be how many or how frequent the disease is. And the distribution is of an event by person, place, and also time. And we are talking about who, when, and when, where, and when. So this is a figure, example figure, showing the distribution of income level and raised plus blood pressure, systolic blood pressure of more than 140, or this diastolic blood pressure of more than 60 in terms of percentages. So you can see that countries with a bigger circle of pink have a high balance of raised blood pressure, and the countries colored with different colors here is actually based on the income level. So this is an example of distribution of disease. And what about determinants? These are factors that present and absent, of which affect the occurrence and level of an event. So usually we ask question of how and why the determinants appears. So these are the figure showing determinants of chronic conditions of NCDs. And we can see that there are a lot of determinants in terms of socioeconomic and environmental risk factors. There are behavioral risk factors, biological risk factors that affects the disease of non-communicable disease. We have heart disease, stroke, cancer, diabetes, and so on. So all these are determinants of chronic conditions of NCDs and explains why a person gets a disease. So let's move on to see on hypothesis testing in epidemiology methodology. So the investigator usually identifies a cause-effect comparison to be tested as the research hypothesis. So the statement should be clear, precise hypothesis, and the study outset ensures that the appropriate data is being collected to answer the research question and avoids the pitfall of drawing spurious conclusions from the data set. So first we ask question and do some background research, then only we construct a hypothesis and test it with an experiment. Okay. If the procedure is working, then we forward to do the data analysis and draw conclusions and look at the results. If it aligns with the hypothesis or it doesn't align with the hypothesis at all. So if it doesn't align with the hypothesis, we go back to the experimental data and construct new hypothesis and retest again. Okay. So if the procedure is not working, then we troubleshoot the procedure and carefully check all the steps and set up and do again the procedures. Okay. Finally, we communicate the results. So a precise hypothesis is important to be set before we conduct the study, before the study outset, okay? So that we get the appropriate data to make a proper conclusion. So why we need nutritional epidemiology studies? Okay, so the main objective of nutritional epidemiology research is to provide the best possible scientific evidence to support an understanding of the role of nutrition in the causes and prevention of ill health. So this contributes to the prevention of disease and improvement of public health by monitoring the food consumption, nutrient intake, and also the nutritional status of a population. Can also generate new hypotheses about diet and disease, can produce evidence that supports or refutes existing hypotheses, and finally, to assess the strength of diet-disease associations. So epidemiology is essential to deliver an effective nutrition programs and also services. We need data before we can even develop nutrition intervention and programs. So the key roles of the community nutritionists include identifying nutritional problems within the community and interpret the scientific literature. And community nutritionists must be able to critically evaluate the scientific literature before formulating new nutrition policies or altering eating pattern messages. So we don't just simply go out there and create policies and create messages to the community. So there are certain elements that can be used to judge the strength of the epidemiologic association and interpreting epidemiologic data. Basic that involves two steps. The first one is to evaluate the criterion for a causal association carefully. The second step is to assess the causal association critically for the presence of bias and also the contribution of chance. So the general principle, the first one is nutrition as an exposure, and the assessment will be from diet, body size, and also physical activity, and the disease as the health outcomes. It can be disease endpoint in terms of cases, number of cases, incidence of cases, and also prevalence of cases that's being self-reported or based on medical records, or we can also refer to disease registries. Second disease endpoint is mortality, okay, which we can look at the vital status registries. Disease also includes communicable and also non-communicable diseases. Example, does a high-fat diet increase the risk of breast cancer? So this is our question, or the question that we want to answer. Okay. Or does a frequent intake of ultra-processed food increase the risk of childhood obesity? So what will be the nutrition exposure and also the health outcomes? So for the first question, the exposure will be high-fat diet. And for the second question, the exposure will be ultra-processed food intake. Okay, so we must measure high-fat diet, whether the diet is high fat, or what is the consumption of or dietary intake of ultra-processed food. And the health outcomes for the first research question is the breast cancer incidence. And the second question is about childhood obesity prevalence. The second general principle. So there are two major types of studies. First is experimental or intervention. The second one is non-experimental or observational studies. So the main difference between these two are the control of research may have over the participants, the methods and the procedures, and also the exposures. So both intervention and also observational studies aims to get causality. So these are the epidemiological study designs. Okay. So first we start with the assignment of exposure by the investigator. Okay. If there is an assignment of exposure by the investigator, so we check whether it's the assignment of exposure is being randomized or not. If it is randomized, then we call it as clinical trial, field trial, or community trial. If the exposure is not randomized, then we call it as intervention trial or quasi-experimental trial. So if the assignment of exposure by investigator is not done, so it can further divide it into two, analytical and descriptive. Analytical is for cohort and case control study, whereas descriptive studies are cross-sectionals looking at prevalence and also ecological studies looking at correlation. Okay? So there are two different types of study design. The first one is experimental study design. The second one is non-experimental, or sometimes people call it as observational study design. The third general principle is there are two levels of nutrition disease relations. First one is at the individual level. Okay? So we can measure this by using cohort, case control studies, and also randomized clinical trial. And the second level of nutrition disease relationships are ecologic or aggregate levels. So these are based on cross-sectional correlations of food data and other prevalence studies, descriptive studies of dietary intake, so on. So this is ecological studies. And general principle number 4 is there are also intermediate endpoints or markers that we can explore on. So basically, this is due to the length of the induction period of the disease. We can't wait. Sometimes we can't wait until the disease appears. So we use markers, okay, as an intermediate endpoints to indicate the disease. Okay. So studies which combine nutritional exposure, intermediate markers, and also disease outcome can be especially informative. So if we have markers as the intermediate endpoints, it can be more informative for us. So can we reliably measure dietary intakes in individuals and population? So when we talk about nutritional epidemiology, so the exposure is about the dietary intake, the food groups, and so on. So can we measure the dietary intakes of individuals and populations? So the quick answer is yes, reliable dietary intake measurement is possible, but it is complex and imperfect. Okay? So there are different methods and each have strengths and limitation, and the accuracy improves when these multiple methods are being used together and when measurement error is properly addressed. So the challenges in measuring diet, because diet is a complex exposure. Okay? People consume many foods in varying combinations and so on. And also diet changes over time, daily, sometimes seasonally or culturally when we have certain festivals and so on, festives and so on. And we will have a time varying data. And there are three error sources in dietary measurement. The first one is random variation, day-to-day or seasonal variation of dietary intake. Systematic error, where there is underreporting, omission of foods, recall bias, and there are also inaccuracy in the food composition tables in terms of incompleteness of the food composition table and also the nutrients that is covered by the food composition table. So this slide here is showing the comparison of major dietary assessment methods. So we have several methods that is commonly used in nutrition epidemiology study. First one is several days or week of diet records or food diaries, multiple 24-hour records, single 24-hour records, food frequency questionnaires, and also biomarkers. Okay? So when we are using several days or week diet records or food diaries, the advantages is high detail and doesn't rely on memory. But it has the disadvantages where it has high participant burden because they need to record all the food intake throughout the day. And this may also alter behavior due to the act of recording. And also the disadvantages is related to food composition limitation. And several days or week diet records or food diaries are usually used for validation studies and also compliance tracking in the intervention. Second method is multiple 24-hour recalls. So these are fairly accurate, less burden than diaries because you don't have to, the participant doesn't have to do the recording, but it will be interviewer assisted. Okay? So we'll get more information by doing interview where we have more detail about the consumption. The disadvantages is recall bias, and it has interviewer burden, so we must train the interviewer, put in some cost for the budget for the interviewer, pay for the honorarium, and so on. And also, the food composition limitation still remains. This method is being used in validation and also compliance tracking in trials. Single 24-hour diet recalls is a less burden than multiple recall because you only meet the participant once, and we can get interviewer assisted detail as well. But the disadvantages is there's recall bias. It needs repetition to reflect usual food. So single 24-hour recalls only describes the food intake the previous day only, not the usual intake. And food composition limitation still remains there. And these are being used in national surveillance of mean population intake, but not at individual level. Food frequency questionnaires are low cost, low burden, and good for long-term diet, and vastly used in epidemiologic research. The disadvantages is there is less detail, and it's very much related to memory based, and the food composition limitation still remains as a disadvantages for this type of method. So food frequency questionnaires are being used in large epidemiologic studies because it can assess past intake and also do trend analysis and also as a validation or validate other methods. The final method is biomarkers, example double label water, urinary nitrogens, and so on. So the advantages is it's an objective, not subject to self-report bias. So the results will depend on the reading of the biomarkers. Okay. And the disadvantages is it's costly and only have few nutrients with good biomarkers. So once you do a test, it's only for certain types of nutrients. The application of biomarkers are usually in the validation studies and also small trials due to its cost. So food composition database is another source of error. Okay? So nutrient values can vary due to season, location, and also cooking methods. So this is especially problematic for nutrients like selenium, where it depends on the soil that the foods are being planted on. And less impact when the goal is only in ranking individuals from low intake to high intake, rather than estimating absolute intake in grams and milligrams and so on. So there are also issues arising from incomplete food composition database and nutrients that are being covered. Okay? Some nutrient food composition database only cover 10 nutrients, while the other nutrient data food composition database might cover 100 types of nutrients. We can use biomarkers to improve accuracy, and also we mentioned previously when we do method combination, we will have better accuracy in determining dietary intake. So biomarkers also help validate self-reports. Okay? So but there's some limitation because of the cost, burden, and also specificity of the biomarkers. So the best method is to use triangulation and validation studies, where we compare the FFQ with biomarkers and also another type of method. And this will improve the confidence of the dietary intake results. So this is meant by triangulation. So we have FFQ here and biomarker and 24-hour diet record to determine what is the true dietary intake. So measurement errors is a recognized issues, but it is not a reason to dismiss the data. So we cannot run away from measurement errors. Okay? So errors are sometimes random, example like in single 24-hour records, and it's sometimes systematic. It's example like social desirability, under-reporting, omission of foods, and so on. So what is the solution? Use multiple 24-hour records or repeated FFQs to reduce random errors. Apply energy adjustment models. Okay? You can also use this method to reduce measurement errors, and also use error correction techniques in statistical analysis. Misinterpretation of data quality is another problem. So critics, like example in Archer et al. Of the enhanced 24-hour diet record in the US misunderstood the key methodological issues of the 24-hour records. So basically, Archer critiques that the 24-hour recall from enhanced is not suitable to assess the population's intake, because single 24-hour recall, as we have seen, have some disadvantages to not present the usual or habitual intake. But national surveys, the aim is to assess population level trends, not just individual precision. So basically, using single 24-hour recalls in the national survey is not wrong, because we are assessing population level trends. Okay? So studies have shown that enhanced methods are reasonably valid, especially after the 2002 improvement on the 24-hour diet recall, where they use multiple pass method, which we have learned previously in nutritional status assessment. 5 steps, you remember. So can we reliably measure dietary intakes? So there is no perfect method, but development and research have led to reasonably valid, reliable methods for assessing dietary intakes. So the tools that support meaningful insights at both individual and population levels good is available nowadays, and ongoing methodological refinement and validation efforts continue to strengthen the field of nutritional epidemiology. Let's talk about inferring causality. So what is the role of nutritional epidemiology in inferring causality? So nutritional epidemiology often use observational data, especially prospective cohort studies, to study diet-disease relationship. So critics argue that observational studies can't prove causality, but randomized controlled trials are often impractical for long-term dietary exposure. So what is the reason RCTs are limited in nutrition field? Because long-term RCTs are expensive and logistically complex, and most of the time is unethical. Nutrients and also dietary patterns are easily randomized like drugs. Exception for nutrients in the pill form, example, folic acid, iron, zinc, etc. And also if we use some short-term biomarker studies. So most of the time we use prospective cohort studies. So these are the strengths of prospective cohort studies. It has a forward-looking design which helps to establish temporality. Prospective cohort studies are also more reliable than retrospective or cross-sectional design. Okay? So it can minimize key biases like reverse causation, recall bias, and also selection bias. So this image here is showing the difference between prospective cohort and also a retrospective cohort. A prospective cohort is a forward-looking design where you start and you follow up to the end of the study. Whereas in retrospective cohort, you meet the participants at the end of the follow-up period, and you try to do a history of the exposure up to the start of the follow-up period. So dealing with confounding factors. So what is confounding? Is an external variable that's related to both the exposure and also the outcome, and it distorts the apparent relationship between them. Confounding factors is a major limitation of observational data, but it can be reduced by collecting data of known confounders, statistical adjustments in regression models. You can also do sensitivity analysis and also do repeated measures over time. But then, if we didn't measure the confounding or unmeasured confounding, can't be fully ruled out in a study. It's because sometimes certain types of confounding factors can't be measured, and those which are not measured cannot be ruled out from the study. So the most popular criteria for checking causality is the Hill's criteria. This is a widely accepted framework for establishing causality in epidemiology observational study, especially in the absence of experimental data. So these are the criteria. The first one is strength of the association. We look at the strength of the association. We look at the consistency of the association across studies. We look at the temporality, the exposure before the outcome, and also the biological gradients in terms of dose-response relationship. Look at the specificity, the biological plausibility, coherence of the study, experimental evidence from previous research, and also analogy. So I've shared a video to understand more of the Hill criteria for consulting. Triangulating evidence. So we have seen triangulation previously. So in triangulating evidence, we use multiple types of data to strengthen the causal inference, where we use prospective cohorts, RCTs or intermediate outcomes, mechanistics and animal studies, and also biomarkers to strengthen the causal inference. Example like for Mediterranean diet and heart disease, before we can establish the relationship between Mediterranean and diet disease, we have done a lot of research in terms of looking at the foods, the nutrients itself, the patterns of intake, and also biomarkers before we can establish the relationship between Mediterranean diet and heart disease. Okay, next, let's look at meta-analysis and systematic reviews. So you have learned about meta-analysis, you know already what is meta-analysis and systematic reviews. So can we trust the findings from meta-analysis and systematic reviews of nutritional epidemiology studies? First, we must acknowledge there are some limitations of meta-analysis and systematic reviews of nutritional epidemiology studies in terms of publication bias, heterogeneity, the study qualities, misleading conclusion due to methodological flaws, misinterpretation of data, and also there is an explosive growth of systematic reviews out there and lack of original data. Let's look at publication bias, where studies with null or negative findings are basically less likely to be published, and those with positive findings will be published in high-impact journals. So this can skew the overall results, as the studies included in the analysis may overrepresent the positive findings. Techniques like Eagers test, stream and field methods can help detect and correct for biases, but the issue remains a significant challenge in meta-analysis. In terms of heterogeneity, so what is heterogeneity? It's the variability of studies between studies in terms of exposure, the outcomes, population, and also the study design. So heterogeneity complicates the pooling of effects, estimates, and can make it more difficult to derive meaningful conclusion. So we need less heterogeneity. There are several strategies that can address this, such as qualitative descriptions, subsetting of studies, and also using random effects models. In terms of study quality, if low quality studies are put together, the overall results might be misleading. It is important for researchers to carefully consider the methodological rigor of the studies involved in a meta-analysis of systematic reviews and potentially exclude low quality studies or report results separately for higher quality studies. Misleading conclusion due to methodological flaws. So systematic reviews and meta-analysis are susceptible to methodological problems, especially when the investigators lack sufficient understanding of the subject matter. So this can result in incomplete literature search, flawed analysis, and also ultimately misleading conclusion. So a key example of misinterpretation of studies on the relationship between BMI and mortality is the conclusion of that overweight individuals have lower all-cause mortality than those with normal BMI, which was found to be partly due to the reverse causation, where individuals in the chronic disease lose weight before death, therefore skewing the results. So when we exclude the individuals revealed with those who have lose weight before death, it revealed that the normal BMI range actually have the lowest mortality rates. Misinterpretation of data. So a lack of familiarity with the nuances of nutritional epidemiology methods can also lead to flawed interpretation. For example, like in a meta-analysis that found no significant association between types of fat, such as saturated or polyunsaturated fat, and also heart disease, actually failed to account for isocaloric substitution of one micronutrient for another. And this misunderstanding can lead to misleading conclusion. The correct interpretation is that saturated fat, when being replaced by refined carbohydrate, do not significantly affect heart disease. But when replaced with polyunsaturated fat, the decreases. The risk decreases. Okay. There is also an explosive growth of systematic reviews nowadays. So the number of systematic reviews and meta-analysis in nutritional epidemiology has skyrocketed, leading to a flood of publication. So these systematic reviews might be beneficial, but also resulted in sloppy reviews and often lacking through thorough evaluation. These reviews are also sometimes rushed to publication by authors who may not fully appreciate the complexity of the issues. And finally, lack of original data. Unlike original studies, systematic reviews and meta-analysis do not generate new data. This can be both a strength and also a limitation. So while these synthesize existing findings, they also rely on the integrity and completeness of the underlying studies. If these studies are being flawed, then the conclusion drawn from them will also be compromised. So the next question we are going to ask is, what is the role of nutritional epidemiology studies in developing policies? First role is to provide scientific evidence to help in developing policies. And also, nutritional epidemiology studies inform dietary guidelines or the development of dietary guidelines. It also supports public health policies. And also, due to nutritional epidemiology studies, we can detect and monitor health risk better and apply the precautionary principles. So nutritional epidemiology studies, especially those with prospective cohort studies and RCTs, provide scientific foundation for dietary recommendation and also health policies. Our Malaysian dietary guidelines are also being developed based on scientific evidences on recent studies on nutritional epidemiology research in the country and from abroad. Although the RCTs with disease endpoints are considered the highest standard of evidence, okay, if you look at the hierarchy of evidence, the RCTs are on the top before meta-analysis. Cohort studies are often relied upon when such trials are not feasible, when RCTs are not feasible due to ethical, financial, and logistical reasons. Example is the USDA or Department of Health and Human Services dietary guidelines are shaped by both cohort study data and also RCTs. Similarly, in Malaysia, our dietary guidelines are also shaped by cohort studies and RCTs from other countries and cross-sectional studies from Malaysian data. National organizations like the NCCFN Malaysia, National Center for Coordinating Food and Nutrition, American Heart Association, American College of Cardiology, US Preventive Services Task Force, and FDA Food Drug Agency use structured grading systems to evaluate the strength of the evidence when developing any recommendation. So we are going to look at some real world examples of policy change driven by epidemiologic data. First one is folate and neural tube defects, or NTDs. So the initial observational studies in the UK in the 1970s highlighted the folate's role in the preventing of neural tube defects. This was followed by case control study, single cohort studies, and also RCTs. And all these studies have shown to support the protective role of folate. And the results is, in 1996, the FDA mandated folate fortification of grain products, which led to a 19% reduction in NTDs within 5 years after the mandate. Let's look at an example on sugar-sweetened beverages, or SSBs. Observational studies have linked SSB consumption to obesity, type 2 diabetes, and also heart diseases. And two large RCTs further confirmed that reducing SSBs intake lowers weight gain in youth. So as a result, various policy proposals and actions have emerged, including the SSB taxes. Example, in Mexico 2013, tax led to a 5% drop in sales. So these SSB taxes are also being implemented in Malaysia. We also have bans in schools and public buildings in cities like Boston. Also in Malaysia, we are banning SSBs in school canteens and also environment near the school. New York City's portion cap also through later, although it's being overturned by in court, was developed based on the epidemiologic research and evidences. Another example is on trans fatty acids, or TFAs, and heart disease. So early epidemiologic evidence have shown, especially from the NERSA study, the link between trans fatty acids intake with increased low density lipoprotein and also heart disease risk. Experimental and also observational studies have shown that labeling regulation by FDA 2003. These observational studies and experimental studies have prompted the labeling regulation by FDA in 2003, local bans of trans fatty acids such as in New York City, and national phase-out efforts culminating in the FDA declaring TFAs as not generally recognized as safe in 2013. So these are all based on experimental and observational studies. So what is the public health impact from this? So it is estimated that hundreds to thousands of ischemic heart disease deaths were prevented annually in places with all these bans. So even in the absence of conclusive RCTs data, observational studies can trigger early public health warnings, highlight emerging risk, prompting precautionary measures, and also help resolve controversies like the total fat intake, omega-3 fatty acids, and so on. So the precautionary principles asserts that if there is potential harm, action can be taken even with incomplete evidence, placing the burden of proof on the industry or scientists to demonstrate the safety of the food products. So observational data also play a role in resolving controversies in nutritional science. For example, there is a debate over the effects of total fat in the diet or the role of omega-6 fatty acids that have been influenced by epidemiologic studies that either confirm or refute the hypothesis counts. So these studies can help policymakers navigate conflicting evidence and also develop more nuanced health recommendations. So when we are developing policies or making change on something, like some regulations, we always need to back up our idea with findings from the nutritional epidemiologic studies. So we cannot just simply wake up on a day and aim to change a policy or make a recommendation on health without any epidemiologic studies to support us. So that's why epidemiologic research is very important, although people might think that why we need epidemiologic studies to be done, why we need another cross-sectional study, another prevalence study, another correlational studies, and so on. So all these epidemiologic studies and data shape ongoing policy debates, and one day we can have a better policy, better health recommendation. So the conclusion from this lecture is, despite there's measurement errors, people will undermine our studies on nutritional epidemiologic data, repeated measures, validated FFQs, and also biomarkers will help improve accuracy. A well-designed cohort studies remain essential for a long-term nutrition research. And combining evidence from cohort studies, where we have a hard endpoints, RCTs, where we have intermediate outcomes, and also mechanistic studies in lab using animal studies, cells, cultures, and so on, leads to causal inference and evidence-based policy. Large-scale international cohorts like EPIC, UK, Biobank, and so on, Malaysian cohort study maybe, will improve methods and deepen understanding of diet-disease relationship. And nutritional epidemiology remains a cornerstone of public health. That's all from me today. Thank you very much.